we do different approach. Sometimes it's an screening method, meaning that I pull tissue of different embryos and then I run arrays. So it gives you a large amount of genes and then we work on that, and with bioinformatics as well. Or another approach is that taking into account all that we know about these animals and the defects, the lineages that we think that are involved, then and all the information available and all the databases that are available online as well, to choose some genes that could be important for the pathway we're involved in the gene that we are interested in. So in this case, I went through literature searching and all the cracker-related genes, I searched through pathways, protein interactions, databases, the genetic interactions, everything available. And I pulled like 120 genes, and then with the gene ontology, I grouped them. I studied very well each one of them, and I grouped them by a signaling pathway. I took into account the expression, where in the embryo, if it is described, where it is expressed. If it is expressed in the tail, probably it's not going to be part of the heart development. So, of course, it's something that's important. I also took into account if it is relevant for heart development, and if the mutant, in case it exists already, shows some heart effect. So from here, I took, it was a little more than that, so like 30 genes, and I ran a quantitative RT-PCR. So with this expression studies, that it was at 10.5 of gestation, some wild type is the blue columns and the knockouts is in orange. And overall, the knockouts are slightly like downregulated. All these genes that you will not be able to read are all important for cardiac development. And something that caught our attention was the ERBB3, that is one gene that is downregulated, as well as ROR2, also downregulated, and are very important and involved in valve defects and ventricular septum defects. This is still ongoing experiments because we always get more genes and more ideas to do. So this is not definitely outside. So when I say that we try to integrate human and mouse, we also do it from the genetic point of view, not only the cardiac diagnosis that we get from the patients. So in this case, one of the students in the lab, Jonathan Chang, I gave my list of genes, that was this 120, he added some others, and he went through all the data that we have, that is whole exome sequencing, from patients that have a POF, or congenital heart disease, versus controls that they have the deletion, but they don't have heart defects. So he did comparison and all the analysis, and he generated these, well, some others also were in the list, but these three are very important for heart development. And one thing, I think the ERBB3 is coincided with the mouse study. So we are going back and forth all the time. I give genes to him. He tries to test them through the bioinformatics approach, and also he gives genes to me, and I go and do expression studies in the mouse. Sometimes it's a qPCR, or sometimes it's in situ. In situ is a technique that lets you to stain the mRNA in the embryo, early embryos. So the conclusions so far are that human endocytosis for crackle have output tract and septation defects. Our allelic series is a good model for human output tract, and it is important because few true knockouts exist in humans. Crackle dosage is in neo embryos leads to a high variation of heart phenotype, and also they are less severe. Probably is the best mimic of the human phenotypes. Crackle neo-minus embryos exhibit a higher penetrance of double outer right ventricle, allowing the use of these allelic combinations to study this specific phenotype that happens in humans very often as well. The phenotypes that we observe in crackle mutants are more varied and involve more than one cardiac lineage. since we have seen muscle defects and it's mainly related to the right side that I didn't mention before, but it is like that. So we are going to think more about the secondary hairpin and not the peripheral field. On the other hand, the valve defect is more related to endocardial lineage, but it has never been described before. And endocardial lineage is part of the peripheral field. And ERBB3, that is part of the epidermal factor signaling pathway, is the gene resulted affected in human and mouse. So I would like to acknowledge from the mouse side that is Morla, my PI Ramis,